All right, is everybody okay with me recording?
Yep, be proud of it.
Thank you.
I was in Pennsylvania for like a few days, and everybody kept asking me, "Are you from Texas?" I'm like, no.
Okay, did you see that I had made an announcement? You've got to get all your stuff in. Okay? All of it. I realized that
That's my daughter.
I had put in the
She is, wow.
in this that for the how I'm working out the grades that 20 points for worksheets. Well, I've only done some for 5 points of extra credit, and I only marked them as each worth being 5 points. So what I'm going to do is I'm going to make them each worth 10 points, and most people did better on the last 2 worksheets. So I'll make those 2 worth 10 points, and the one original one you did, that'll be worth 5 points, and that can be the extra credit.
Did you grade this already?
The worksheets?
No.
Yeah?
I have.
Mine's posted.
I've posted them. If you handed them in late, I didn't do anything else yesterday. It took me all day.
Wait, we took them in Monday? Is that like?
Yeah, well, it actually was late. Yeah, I didn't do the worksheets because I know that you wanted your exams and your art projects. Okay, I didn't have time for anything else, plus preparing a review for you guys. Yeah.
I turned in both the worksheets originally, but then you said it changed something.
Yeah, so I've graded one of yours but not the other.
Yeah, I forgot to give it back after that.
What, you forgot to what?
I forgot to give it back after
So give it to me now. Now you want to give it to me, don't you? I think that's all I need. Do I have any other? Don't worry, I'll work everything out to everyone's best advantage. Or I'll try to do that. I had the worksheets all graded, and was ready to hand them back so that you'd know what you did wrong before the exam. But it took, because of the problem with the internet, it took me all that time to. I don't think I bought them this time. Did I bring them? Unknown, NMR. No, didn't bring them this time. Sorry. But if anybody asks me, I can tell you what you did wrong. Oh, no, because somebody's still handing it in. Better not do that then, because then he can cheat, can't he? It's not fair. Okay, so Dolphin's not here. Michaela, Nikki, Chance, Edie, Michelle, Haley, Stephanie? No, not yet. Chris, Haley, Mary Hannah, Ebony, Jenna, yep. Eric, there we go. Okay, I just can't wait to spend 2 hours writing on the board. So we're going back to scratch. Are most bacteria and fungi pathogenic? Exactly. Did everybody get that? Most fungi and fungi and bacteria are not pathogenic, are they?
Nope.
There's only a few that are.
I'm gonna record.
Is everybody happy with Chris recording?
Yep.
No.
Well, you can leave.
Okay. Koch's postulates. God, you guys have heard this so many times. Koch's postulates. They were experiments originally done on Bacillus anthracis, right? Or I think you pronounce it Koch now because, the American way is to pronounce it Koch. You know, that's the politically correct way.
Coke, those guys named the Koch brothers.
Yeah, well, they're bad. Unless you're a Republican. I'm not a Republican, so they're bad.
They're all bad.
Yeah. They're trying to manipulate the oil market. Okay, so Robert Koch. He did these experiments on Bacillus anthracis. And what's the basis of them? What do they do?
To prove that one thing is the cause of the disease. That one
Right.
thing.
Exactly. That's the basis of them. That microbes or microorganisms cause disease. That's the basis of them. And there's 4. Do I need to go through all 4? Just know them, okay? The first one is you have to be able to isolate the disease-causing microorganism from the animal that has the disease. And you have to grow it in pure culture. And then you have to be able to inject that into a healthy animal, and that animal has to get the same disease, right? And then you have to be able to isolate the same microorganism from the second animal that got sick. You know, he's the one who was sacrificed for altruistic reasons. And you have to you have to be able to grow that one in pure culture too. And you can't do Koch's postulates these days. That was one of the questions. For ethical reasons, a lot of times. They can't do Koch's postulates for AIDS, can they? They do it with monkeys, but not with humans. The humans might get a wee bit resentful, apart from the fact that they might die. Okay, did you get all that? Louis Pasteur showed that microorganisms are present in the air, right? And he was the one who devised pasteurization, which reduces, by heating, he showed that by heating you could reduce the number of bacteria, and what it does is it kills most of the pathogenic bacteria. So that's what pasteurization does. It kills most of the pathogenic. But it's not sterilization, right? Sterilization is killing everything. Okay. Now we're going to go right back to chemistry. Aren't you all excited about that? Enzymes. What do enzymes do?
Speed things up, right?
What's that?
How to speed things up.
Yeah. How do they do that? They speed up chemical reactions, right? And how do they do that?
Lower the energy of activation.
Right. Did you everybody hear that? They lower the activation energy. They're biological catalysts. They are not used up in the chemical reactions. They're not affected by the chemical reactions. They just cause the chemical reactions to occur. Okay? Enzymes are all proteins. Except for ribozymes. They're RNA. Yeah.
Quick question, have you seen the final?
Yep.
Yeah, how long is it?
80 questions, which you should easily be able to do in 2 hours, because I normally give you 60 questions that you can easily do in an hour and 15 minutes.
Do we come here or do we go to
The testing centre.
Okay, so
Hopefully.
we go on the computer?
Yeah, hopefully. Hopefully I won't have to give you a hard copy again. But it won't be so bad this time, because last time I got an hour and a half or something to make you a hard copy. So I was somewhat stressed. So if there's, this, you don't have to worry if there's strings of things and stuff like that. I didn't have time to even look at that for the last exam. I know there was a string of D's.
There sure was. About 8 of them.
Yeah. But do you think I had time for that? I got that done with 1 minute to spare.
I always hate when that happens.
Yeah.
It makes me so
Awful. Yeah.
I was like, look at that.
So don't worry about anything like that. Just focus on the answer to the question. Forget any of that psychological shit. Stuff. Okay. Chemical bonds. What are the 4 types of chemical bonds? No, 3 main types, right? Okay. We start out ionic. Okay. Covalent. And hydrogen, right? Ionic bonds are when, I should get my pointer out. Remember how I told you elements, they want to have their outer shells, electron shells full, right? So the number of electrons in each shell is, you can tell by the rows, okay? So the first shell has 2 electrons in it, the second shell has 8. But all you have to worry about is the outer shell. So if they have 1 electron in the outer shell, then they'll lose it to have a full shell, right? If they are missing 1, then they'll gain an electron. So if they lose an electron, then they have remember there's the same number of protons as electrons. In a normal atom, there's the same number of protons as electrons because they have to be neutral. So if they lose an electron, then they have a charge of +1, right? Because they have more protons than electrons. If they gain an electron, then they have a charge, where's the eraser? Of -1. -1, okay? Now this thing that has a charge of +1 is attracted to the thing that has a charge of -1 because opposite charges attract, right? So when if it loses an electron or gains an electron, or it is called an ion, right? And this is not like what you iron your clothes with. And then these things that are attracted to each other, they become close. That's an ionic bond. Okay? It's the attraction between ions of opposite charges. Yes?
It's always a metal and non-metal, right?
Yeah, it usually is. So, for example, lithium usually becomes positive. All these ones down here will become positively charged, and all these ones down here will become negatively charged because they have 1 electron to add. So the molecule, each atom becomes close to each other, okay? So that's a reasonably strong bond, but the covalent bond is when they can lose, it takes energy to lose or gain an electron. So to lose 1 or gain 1 electron doesn't require that much energy. To lose 2 electrons or gain 2 electrons requires more energy, and you can lose or gain 3 electrons, but you cannot lose more than that. It just requires too much energy. But they still, crave, those atoms still want to have their outside shell full. So when it's past that, they share. They share with something else, and that's what a covalent bond is. And in order to share, say you have, this is carbon, has 4 electrons in its outer shell, okay? So it shares with 4 other things, like for example hydrogen, which has 1 electron. But when they share, their electron clouds actually overlap. So this is hydrogen here, and this is carbon. See? So their electron clouds are overlapping. So it's a very strong bond, and they're very close together. Can everybody see that? So this is the strongest bond. This is the shearing, and this is the strongest. And remember hydrogen, so this is what oxygen. Oh, also remember when two electrons are shared, it's represented in chemistry by a single line. Okay? So this is methane here. This would be written by like that. And carbon is unique in that it can, because of its 4 electrons on the outer shell, it can form 4 covalent bonds, and because of that it can make huge molecules. You know, it can have strings up here, strings here, strings here, so you can have huge molecules. This is water, right? H2O, and you have 2 electrons shared here, because oxygen has 6 electrons in its outer shell. So it shares 1 electron with hydrogen, with each of these 2 hydrogen atoms. And what happens is, remember oxygen's greedy, because it's much bigger and has more electrons. So it keeps those electrons closer to it. So the oxygen part of water becomes slightly negatively charged, and the hydrogen part becomes slightly positive. Those funny looking D's mean slightly. And because of that, so these hydrogen bonds are slight, weak bonds between molecules that have oxygen in them, and hydrogen. Okay? So that's the bonds that are between DNA, right? So these are the weakest bonds. These are the hydrogen bonds. So this is the strongest, covalent. The next strongest is ionic, and the next is hydrogen. Hydrogen bonds are between molecules. But then within the water molecule, so between water molecules is hydrogen bonds, within the water molecule is covalent bonds. Does everybody get that?
One more time.
Within, inside the water molecule is covalent bonds. Between water molecules, because you can see the water molecules, that's what makes water form a liquid, is hydrogen bonds. Between, because the oxygen from the next water molecule will bind here, oxygen from another water molecule, etc. They line up. Most of the bonds in organisms are covalent bonds. We're made up of, our body's made up of covalent bonds. That's what holds us together, right? Hydrogen bonds are what hold the strands of DNA together, right? The nucleotides, on the nitrogenous bases are held together by hydrogen bonds. Hydrogen bonds you can break by heating, and then they'll come back together again. Because water, you heat it up, evaporates, comes apart. Yes?
That's why the sticky ends works, right? Because they have to form another hydrogen bond.
Yeah. Yeah. You can break them apart with, those enzymes break them apart, but you can also break them apart by heating. That's what you do when you do southern blotting and all of that kind of stuff. Yeah.
Do we have to know any functional groups?
No, I don't think so. You might have to know amino acid. Yeah.
So covalent's DNA.
What's that?
You say covalent with DNA?
No, hydrogen bonds between the strands within the molecule is covalent. Yeah. Between the 2 strands of DNA. And then there are actual hydrophobic bonds. Hydrophobic bonds are between long molecules like this that have a whole bunch of carbon and hydrogen, like fats. And the way to think about hydrophobic, they're not actually bonds, they're interactions. The way to think about those is, if you put oil in water, what does it do? It all, globs together, doesn't it? Because they like to be next to each other. So and they resist water, right? And that's lipids are an example of hydrophobic interactions. Hydrophobic just means water hating. Okay. There's two types of compounds. Remember, a compound is a molecule made up of more than one atom, and they're different, right? So inorganic compounds lack carbon. And water is an inorganic compound, right? Because it doesn't have any carbon in it. And this, because it's considered a polar molecule, see, because it has poles. You know, this part's positive and this part's negative. And they line up according to the poles of the molecule, molecules. Organic molecules always contain, what do organic molecules always contain? Carbon. And what else? Because carbon dioxide is inorganic, isn't it? So they have to contain carbon and hydrogen. But definitely carbon, okay? And as I said, they can become very structurally complex, because you know how complex we are. Okay, the pH. Everybody loves pH. What do I do with all my pens? Left with that crummy green one.
The black one.
One's on the ground.
Oh, the black one's on the ground. No wonder I can't find it.
The green one's on the shelf.
Yeah, but the green one's crummy.
Um,
It's one of those. Okay. pH. pH goes from 2 to 14, right? The lower the pH, the higher the hydrogen ion concentration. So it's the opposite. Low with pH, higher H plus concentration, hydrogen ion concentration. Don't be confused about that. So a pH of 2, and a pH of 4, which is more acidic? 2. Which has the higher hydrogen ion concentration? 2. Has everybody got that? How much difference between them? This is how much more acidic than this? 100. Yeah, because each division in the pH is 10 times. Everybody got that? So 2 divisions, because this is a logarithmic scale, remember? P means minus the log of. So pH equals minus the log of hydrogen ion concentration. So you can tell it's the opposite because it's minus. So don't forget that. Everybody got that? They're opposite. Just like the BOD and the oxygen concentration in sewage, right? In water. The higher the BOD, the more sewage, and the lower the oxygen concentration. I always remember it now the way Chris remembered it. BOD, the D, dump. It's good. It was good. Good analogy.
in bases, what is it that they have more of than in acids? Is it OH?
I was going to tell you. Yeah. Bases, so the pH of 14 would have a much higher hydroxyl concentration. But I don't think they're going to ask you that. I think you're just going to need to know about hydrogen ion. Okay? So a pH of 14 and a pH of 11, this would be 1,000 times higher hydrogen ion concentration than this, right? Can everybody see that?
I don't get that at all.
Okay.
More acidic.
This one's, yeah, 1,000 times more acidic. Okay, you've got to subtract this from this.
Okay.
Okay, that's 3.
It's a little, okay.
3, right?
Okay.
And 3, that's how many times you've got to multiply 10. So the 3 is what power you've got to put 10 to.
Okay. Remember the little thing, like times 10, times 10, times 10, had like counting? It was like a.
Yeah.
So 14 minus 11.
So the difference you got to put as a power, because this is log of pH. This P means log of, so it's your exponent.
Okay.
Okay, do you get it now? Yeah. So when you're subtracting these, you're actually subtracting exponents.
Sorry.
There you go. Okay. Does everybody get that? Okay. This acids dissolve into hydrogen ions and bases and negative ions. Negative ions are anions. So a negative ion is an anion. A positive ion, I just remembered this by remembering what the positive was. A positive is a cat. So positive ions are cations. So I like cats, so I'm positive about cats. So the positive one is the cation. So they're the positively charged ones,
My
right?
teacher always drew like the T and the cation, like really big.
Oh.
That looked like a plus sign.
Oh, that's clever. T like this.
Like C-A-T.
Oh, C-A-T. Very good.
Like a plus like that.
Yeah, see, and that's a plus. Yeah, that's clever. I didn't worry about the others, and I just remembered that the cation was the positive one, and I love cats, so they're the good ones. So, but that's clever. Okay, we did 4 types of organic molecules, right? You need to know the monomer of each, okay? So we started off with polysaccharides, right? Polysaccharides all have the same formula. C6H12, oh that's O6, that's glucose. So they have twice the number of hydrogens as carbons and oxygens, so they all have the same formula.
CH2O.
Yeah, CH2O. So, like glucose is C6H12O6. So it's always the same, twice the number of hydrogens as there are carbons and oxygens. Everybody got that? So that's carbohydrates. So what is the monomer of carbohydrates? Carbohydrates are sugar molecules, right? Carbs, everybody. Carbs are what makes you get fat, gives you energy. What's the monomer? What's the unit that makes them up? It looks like this. It's a ring structure. Whoops. Monosaccharide! Because carbohydrates are polysaccharides, right? An example of a monosaccharide is glucose or fructose. Flour is just a whole bunch of glucose molecules joined together. And the glucose is the monosaccharide. Everybody got that? Has anybody not got that?
Polysaccharide. So what's a polysaccharide? Oh, yeah, example of a-
Starch,
Okay.
glycogen.
Okay.
Sometimes, depending on the branching, whether they're sweet or not. If they have a lot of branching like flour, then they're not sweet. Cellulose is another polysaccharide. Okay. Lipids. They have the same formula, but it's not with the same arrangement. So they're composed of carbon, hydrogen, and oxygen, but not in the same ratio. Remember there, those big long molecules? What do I do with my black one again?
It's by the chair.
Oh. See, I need you guys. Lipids. Everybody knows what lipids are. Those are the things you hate to put on weight with, aren't they? And they look gross. Evidently, you know, the surgeons residents that I used to work with used to tell me about doing surgery on obese people. And they said your hands get all covered in fat. Gross. Puts you off eating, doesn't it? Your hands get all greasy and everything. Okay, so remember these are these strings of C's. And then they have that, so that's a fatty acid, a whole bunch of C's with H's here. Okay, that's a fatty acid, and it has, I don't remember what the acid is. C double bond OOH. That's the acid part. Okay, and then you have triglycerides, which glycerol is this here, and you have string here. So for phospholipids, which are part of the membrane, you have a phosphate group here.
Makes it hydrophilic.
Yeah, that's because the phosphates are negatively charged. And then you have the two, so they're like two, phospholipids have two sets of fatty acids. So I just told you what the monomer of lipids is. What's the monomer of lipids?
Fatty acids.
Yeah. And they're just basically strings of carbon and hydrogen with some oxygen up here, because glycerol is like this. Okay, you don't have to remember what they look like, you just need to remember the monomers and what they are, okay? And you know what? In phospholipids, what are phospholipids part of? What are they for? Cell membrane. Cell membrane. Phospholipids, cell membrane.
And a phospholipid is
Yeah. Remember they go like this. Phospholipids self-assemble. They form membranes. They just naturally do that. Because this part is where the phosphate is, and in water, the phosphate part is attracted to water, and these carbohydrate parts don't like water. They're hydrophobic, water hating, so they face away from the water. So if you put phospholipids, a whole bunch of phospholipids into water, it would naturally form a membrane type cell. Okay? They just do it. Proteins. What's the monomer of proteins?
amino acids.
Yay! And this is what an amino acid looks like. Everybody should remember this. God, can I remember that? Oh, I've got it here. Amino acid.
Is that an A or an H on the top corner there, on the left-hand side?
H.
H. That's the amino group. Remember the functional group? And if you cut that off, you know, during degradation, you get ammonia, right? That's the next part after releasing the amino acids, which was a question in the exam, right? And this here is a carboxylic acid group. So this is the acid part. Now each amino acid, there's 21 of them, each one has a different group here. And one of those groups is SH. And that's the only reason we need sulfur, because these SHs form disulfide bridges which hold the secondary and tertiary structure of proteins together. Right? Everybody damn well should have got that question. It's the only reason we need sulfur. But you know that we all need sulfur, and we've all eaten sulfur, because when we die and we've been eaten up by bacteria, you can smell the smell of the hydrogen sulfide, right? That's the rotten egg smell. Okay, now remember that there's 4 levels of protein structure, right? The primary structure is...
just the sequence of the amino acids.
Right. And the primary structure determines everything else. What determines the primary structure of the amino acids?
DNA.
Yep, the sequence of the nucleotides in a gene. Okay. See, when I first asked you guys that, you didn't know it, you see, and now you're all knowledgeable. The secondary structure. Secondary structure is the beta-pleated sheets or the alpha helices. So you have this sort of thing or this. That's the secondary structure. What holds these together? Yeah, but what's the other thing that holds them together?
Hydrogen bonds.
Hydrogen bonds, yes. That's the main thing, hydrogen bonds. But obviously we need sulfur as well. We need the disulfide bridges. And the tertiary structure is when it, you get all this. You get some alpha helices, and then you get some beta-pleated sheets. And then the little pockets are what forms the active site on the enzyme. Because proteins are enzymes. So say like this is the active site where the substrate binds. So the active site, the little pocket, is extremely important because that's how it can control all the reactions that occur in a cell. So enzymes control all the reactions that occur in a cell, and enzymes are made, the structure of enzymes are determined from our DNA, and that's how our DNA, our genes, control everything that's going on in the cell. Does everybody get that? It all comes back to the brain of the cell, which is the DNA. Okay, and the quaternary structure, what's that? When you have more than one subunit of a protein. Not all of them have quaternary structures. The classic one is hemoglobin. It has 4 subunits, joined together by an iron molecule.
I don't know, can you repeat that again?
The quaternary structure, it's when you have more than one subunit of a protein. Okay? And the classic is hemoglobin, which is 4 subunits exactly the same, held together by an iron molecule. And the iron molecule is what carries the oxygen. It's what binds to the oxygen. Okay. Nucleic acids. See, the green doesn't erase very well either. It's really crappy. Nucleic acids. They all have this structure. This is my nitrogenous base. And this is CH2O. Okay. And okay, this is the nitrogenous base. Oh yeah, so what is the monomer of nucleic acids? Yeah, and what are the 2 types of nucleic acids? No, nucleic acids. RNA and DNA.
Yeah.
Yeah.
You just say, isn't that exactly?
You were starting to say that. I'm sorry, I interrupted you. Okay. So, and the monomer is the nucleotide, and the nucleotide has this basic structure, right? Where the phosphate is the 5' end, because you remember each corner in one of these ring structures represents a carbon atom, unless something else is put in there. So there's a carbon here, there's a carbon here, there's a carbon here, and there's a carbon here. You number carbon atoms from this side, left side. I'm not very good with left and right. So this is number 1, this is number 2, this is number 3, this is number 4, and this is number 5. So even though it's actually the phosphate that you're talking about, they call it the 5' end. So the 3' end is here, where this hydroxyl is. This is the ribose or deoxyribose. Right now it's deoxyribose because there's no OH group here. It's just an H. Okay? So this is the sugar molecule. And this is the phosphate. So it always goes base, sugar, phosphate. Okay? This is where hydrogen bonds to the other. And the other strand goes in the opposite direction in DNA. Remember? So its ribose is up here. Because it's anti-parallel. Okay. You have to know which base bonds to which. So, C bonds to what? G. There's actually 3 hydrogen bonds there. So that's cytosine and guanine, and adenine to thymine. Thymine is in DNA, right? In RNA it is? Yeah. Okay. Now, for DNA replication, RNA transcription, and for protein synthesis, for translation, remember, everything goes in the 5' to 3' direction. Okay? Everything is 5' to 3. Got to remember that. Okay.
What was that for?
For DNA replication, RNA transcription, and translation, RNA translation, all done in the 5' to 3' direction. Proteins, we'll talk about replication first. DNA replication. You're lucky, I'm just telling you the very basics. Yes?
Can you do me a favor and on the picture where you have it starting to bind to the other DNA molecule, can you draw the end that it binds to? Because I keep getting confused on that.
I don't want to do that. I've got to do it upside down. So it would be so this is if this is adenine, this is thymine, and you've got to do it the opposite way round. The phosphate. Phosphate is got to come 5'. So this would be.
This is mean.
I mean, no, it's really mean
You
too.
need to draw it there. Look it up online.
Yeah.
You have a phone.
Okay, you happy now?
Yes, very happy, thank you.
So they're like this. Bloody well better be. Okay. Okay, during DNA replication, you have to copy 2 strands, right? So you have your 2 strands of DNA, right? And this one is the 5 to 3 prime. And remember, because they're in opposite directions. Okay, so because it's going 5 to 3, so this would be the 5 prime end of the one that's being copied. The leading strand goes in one piece. Remember, DNA polymerase, both strands are replicated in the same direction, but because the other strand is in the opposite direction, the lagging strand, so this is the lagging strand.
So the bottom strand's lagging.
Yeah, I think so. But you got the general idea. It has to go like this. So it's still going forward, but
Wait.
it's going forward by going backwards. The DNA polymerase,
But I thought it was the other way. I thought it went 5 prime to 3 prime, not 3 prime to 5 prime.
it's going 5 prime to
okay,
3
okay,
prime.
Okay, I gotcha.
5
It's
prime to 3
copying
prime.
it.
It reads backwards.
So when it's copying, it's 5, like when it's, okay.
Yeah.
Gotcha. Okay, that makes sense now.
This is the leading strand, right? This is the lagging strand, but they're still, because the replication fork goes in the same direction. But this one, because the strand is opposite, it has to do it in little pieces, but it's still going forward. It's going forward by going backwards. Do you see what I mean? And what are these little pieces called?
Sticky ends. Fragments.
Yeah, named after, I think he's a Japanese guy. What would you say? Wouldn't you say?
Yeah.
Sounds German.
Okazaki fragments. So if you look at you can actually label the DNA and you can see this happening. If you see a DNA being copied, a plasmid being copied, you'll actually see it starts here and you'll see the replication forks going in opposite directions. You'll see this. Because the 2 strands have to be unwound, pulled apart. So this one's going in this direction and this one's going in this direction. But one piece will be going like this, and this piece will be going like this. Do you see? But both pieces are going in the same direction when it's being copied. Okay, so that's DNA replication. The enzyme that copies DNA into DNA is called DNA polymerase.
Okay, that does what and what? Say that one more time.
DNA into DNA. They're always called polymerases because they make polymers, big long strands of DNA. Okay, that's DNA to DNA. And that process is called replication. Okay? Replication has to occur before a cell can divide because you have to make another copy of your DNA to put into your daughter cell, right? Okay. I'm really exhausted. So, this DNA, in order to be made into protein, can I erase this bloody thing?
Just don't
Wait,
erase
no,
the
no,
cat.
no.
Don't erase the cat.
Draw it one more time for Mitchell.
You guys have been mean to me.
We're trying to keep our spirits up.
Okay, so this is the hardest part. Hopefully once we're done with this it won't be so hard. Okay, protein synthesis. The template for protein synthesis, what is the template? Yeah. The template is the gene. It's the DNA. Okay? So the template is the DNA. The sequence of nucleotides in the DNA that make up the gene, right? That's the template. And protein, in order to get this to the protein stage, first of all what happens is it's transcribed, right? So you have the process of transcription. What does the process of transcription involve? Copying DNA to RNA. That's transcription, right? And what enzyme does that? RNA polymerase. Right? So your DNA is copied into what's called messenger RNA. The messenger RNA is the messenger that carries it from the nucleus out into the cytoplasm. And where does it go? Where does the messenger RNA go? In order for that message that's on the messenger RNA, it has to go to the ribosomes. And at the ribosomes, what happens?
Translation.
Yay! It's translated. Translation. So that process is called translation.
So,
It's translated into protein.
so the mRNA gets translated into protein?
Yeah, at the ribosome.
Why does it seem so much simpler now?
Because you're so much smarter now, because you've been working with me for so long. You know the two ribosomal subunits, they bind together on the ribosome, I mean on the messenger RNA, which is single-stranded, one string, and they move along it. They move along it, and as they move along it, a new tRNA is put in, and each tRNA is bound to a single amino acid.
So one tRNA for one amino.
Yeah. And each tRNA, the tRNAs have on them, because the message, how is the message written? On the messenger RNA, the gene is written as codons.
Okay. Well, what did you say?
Codon. And each codon is composed of,
3 bases.
yeah, 3 nucleotides, 3 bases, each codon, right? You need to know how, remember that chart? Do you remember how you need to be able to read that? Okay.
Can you draw the chart for me?
No! Mitchell's being so mean. The chart, there's an animation that tells you exactly how to do it.
You have that on Canvas, right?
Yeah. Remember, for example, C-A-G. Remember you read the first one, the second, and the third. There are 3 stop codons, and there's 1 start codon. The start codon is AUG. I can tell you that now. Sorry, that's meant to be a U. And the reason it's U and not T is because it's RNA, not DNA, right?
So that's a start.
Yeah, that's the start codon. You don't have to know that. You have to be able to read the
Oh,
chart.
that's that whole thing.
Yeah. So, yeah, remember that there's more than one codon for some amino acids. Okay, the tRNAs have on them, what do they have on them? The anticodon. Remember that tRNAs have this structure? And right here is the anticodon. Right? What am I doing wrong?
Nothing.
So if the codon is CAG, oh, it actually wouldn't be CA. Oh, yes, it would. the anticodon would be GUC, right? Yeah. Right? And each tRNA only binds to a specific amino acid. So the anticodon determines what, on the tRNA determines what amino acid it's going to bind to. Does everybody understand how translation works? Go on.
There are so many really helpful things on the internet.
I just confused
It's
myself.
really simple.
Okay. Protein synthesis. Another name for protein synthesis. Protein synthesis involves transcription and translation. Another name for that is gene expression. Because you're expressing your gene into a protein. Okay, the tRNAs, these are going to be 3 to 5 prime. Okay, the tRNA. When your DNA is transcribed, this is the 5 to 3 prime end, and this is the 3 to 5. The strand that is transcribed is the 3 to 5 prime one. Okay? So the messenger RNA, this piece, is going to be 5 to 3 prime. Because remember, everything has to be 5 to 3. So the template that's actually transcribed is the 3 to 5 prime strand of your DNA. Does everybody see that?
So you end up with a 5 to 3, because that's what you want.
Because the messenger RNA is 5 to 3.
Okay.
Remember, because everything has to go in the 5 to 3 prime direction. So this messenger RNA is being made in that way. Okay? So it's this strand that's copied. Okay? So the anticodon would be in the 3 to 5 prime direction. So the sequence of the anticodon would actually be the same sequence as the template. Can everybody see that? You're going to have to think, which sequence is which. Okay, but by convention, when you write a sequence of DNA, you never write both strands, because that would take too long. Remember when we gave you the sequences for the BLAST and everything? You only write the 5 to 3 prime sequence. Okay? So whenever you report anything, you always report that 5 to 3 prime direction, not the 3 to 5 prime. Okay? So if I'm asking you for the sequence of a sticky end, so for example, you have the sequence GAATTC. Okay? That's obviously a palindromic sequence. So the other strand, you know what the other strand would be, right? This is the 5 to 3 prime. You're only ever given that sequence because you know the other sequence would be this, right? Whoops. Right? This is the 3 to 5 prime direction. Okay, if you digest that with a restriction enzyme, say this restriction enzyme cuts here. If it cuts here, you know it's going to cut here on the other strand. The sticky ends that you would get would be this. This would be this end. So this is 5 and this is 3. And on the other side, you'd get this. Can everybody see that? So this is going to stick to this, right? So say you get asked, say you get this. This is 5 prime, 3 prime. And this is 5 prime here. And it asks you what would be the complementary sticky end that would bind to that. What would you write? you'd write TTCAA, right? TTAA. If you were just given that, what would stick to that? TTAA, right? AATT would stick to this. Can you see?
Yeah.
They're clicking.
They're swapping out.
Yeah. But that's the 3 to 5 prime, isn't it?
So you gotta write it the other way.
Yeah, you've got to write it the opposite way. So your answer would actually be AATT. Can everybody see that?
This is stupid.
Because you never write sequences in the 3 to 5 prime direction.
So if it asks that, it's just gonna, like, the answer is gonna be,
The same.
okay.
It's going to be the same because it has to be in the 5 to 3 prime direction.
So the answer's already given, you just had to know that.
Yeah, you have to know that by convention sequences are always written in the 5 to 3 prime direction. Can everybody see that? It's got to get easier. Metabolism. So, we've already talked about enzymes. They catalyze all the reactions, right? In metabolism. It's the enzymes that do that, but they don't get used up themselves. Metabolism consists of catabolism and anabolism. Catabolism is the breaking apart of molecules into their monomers, right? And then they break them into even smaller things than that. Okay. Anabolism is the building up of smaller molecules into bigger molecules. The joining together of monomers to make a bigger molecule, what type of reaction is that? Yes! Hooray! Dehydration synthesis.
Is what one?
Joining of smaller molecules to make bigger molecules, because you lose water in the process. Condensation is another name for it. Okay? And the breaking apart, because it requires water, that's called hydrolysis. And this is dehydration synthesis. So you're remembering some things. Okay. Glucose catabolism. What's the first step? There's 3 parts to it. The first part is glycolysis. Am I waking you up?
What is this?
Glucose catabolism.
Mitchell needs visuals.
Mitchell needs visuals. Can you draw the visual one more time?
Bloody Mitchell.
That was so adorable in your accent.
That's what we say in New Zealand. Okay. So the first part is glycolysis, right? It doesn't require oxygen, does it? And what are the products of glycolysis? No, that's the next step. What's that?
Wait, what did you say?
He said acetyl-CoA. Yes! Pyruvic acid or pyruvate. Just remember the pyruve part in your set.
Glycolysis product is
Pyruvic acid and ATP, and you get some NADH made too. But this is the main purpose of glycolysis is to make pyruvate. You only get a net gain of 2 ATPs, right? Because you use 2 ATPs. and you get 2 pyruvates, right? Which are 3 carbon atoms. This is a 6 carbon atom, and you get 2 3 carbons. Okay. Now, if there's no oxygen, you can stop at this stage, okay? And if you stop at this stage and just convert this NADH back to NAD,
That's fermentation.
yay! Fermentation. And one of the products of fermentation is
Some gas. Lactic acid.
yes. But everybody's forgetting the one that yes, alcohol. Ethanol. That's the shorthand way of writing ethanol.
Anaerobic acid.
Yeah.
Okay, so NADH goes to NAD.
Yeah. Now remember, okay, you have to know oil rig for this. Reduction.
Is gain oil or
Yeah, oil rig is oxidation is loss of electrons, reduction is gain of electrons. But remember in organic chemistry or in biology, you watch the hydrogens. So when it loses a hydrogen, it's oxidized. When it gains a hydrogen, it's reduced. So this is the reduced form, right? NADH is the reduced form. The other, these are coenzymes. The other one that is used to carry electrons is FADH2. So they're both reduced when they've got hydrogens on them. Okay? Now, you can see it happening up until the electron transport chain, and then you can't. Then it's electrons that are being passed. Okay, the Krebs cycle. This only occurs in bacteria that can respire. So remember, you can have 2 types of respiration. You can have anaerobic respiration. That's when oxygen is not the final electron acceptor.
And then, but
It-
doesn't it just stop?
No, not if they've got something that will accept the act as the final electron acceptor. Okay, if it normally, like us, we require oxygen to be our final electron acceptor. If there's no oxygen, then the whole lot shuts down. We die without oxygen, right? So if you get asked a question like that, just think about yourselves. You rely on aerobic. We are totally aerobic, aren't we? Without oxygen, we're dead meat. So, oxygen as the final electron acceptor, that's aerobic respiration. And that's the most efficient. You get the most ATP from aerobic respiration. Anaerobic respiration is the next best. And then the worst is fermentation.
So aerobic is the best?
Yeah. We should know that because we're the most efficient, aren't we?
How many?
Hmm?
You said how many ATPs was from the oxygen, from aerobic?
I think there's like 38. That's when everything goes great. 36 in people. 38 in bacteria. I have to sit down for it. No, because two comes from transporting across from the mitochondria. And you know in aerobic respiration that glycolysis always occurs in the cytoplasm, right? In both. Glycolysis in the cytoplasm. In eukaryotes, Krebs cycle and electron transport in the mitochondria. In prokaryotes, which are bacteria, the Krebs cycle occurs in the cytoplasm, and then the electron transport chain in the plasma membrane. Okay, we got that? Okay, the Krebs cycle. What's the main purpose of the Krebs cycle? You had a question of this in your exam. Yeah, to produce NADH and FADH2 to lead into the electron transport chain. It provides the electrons for the electron transport chain, and they're carried as hydrogen ions, right? As hydrogens. Okay. The Krebs cycle, the intermediate step which makes acetyl-CoA, and the Krebs cycle produces all the carbon dioxide. All the carbon atoms from the glucose molecule, which is 6, are given out as carbon dioxide. Okay, it all occurs in the Krebs cycle, that part. But not much ATP is made in the Krebs cycle. Okay, and then from the Krebs cycle, the NADH and FADH2 goes into the electron transport chain. And it's a series of reactions, they're called cytochromes, that pass electrons from one to another, and at each stage ATP is made. From NADH2 you get 3 ATPs for every NADH2. For FADH2 you get 2 ATPs. Now in bacteria, the electron transport chain doesn't always make ATP. What is it used for? It's used to make a gradient, remember, like a battery. And that battery is used to, yeah, it's the battery it's the power for their outboard motor. Remember, you have hydrogens pumped onto one side. So you have your membrane like this, and it pumps hydrogens to the outside, and there's negative charges on the inside. So you have a battery here, and it lets in those hydrogens at the same time as making the flagella go around. Bacteria have a corkscrew flagella, remember? Not like eukaryotic flagella that whip. Somebody should know about flagella very well because they did a project on that, art project.
She's not here.
Oh, she's not here?
Nikki.
Nikki, yeah. She was hoping to come.
Whoever's recording the audio of this, can you load it onto?
Yeah, it takes like 2 days, so just give me time because it takes forever.
Don't you sacrifice your studying in order to do this?
My computer does it. I mean, I can listen to it well, but it just takes, you have to upload it into a movie maker, and then the movie maker has to save it, and you have to do it on YouTube. It's ridiculous. I'll do it though, don't worry.
Isn't she nice?
Yes.
Okay.
So is the chemistry the longest section of the review?
It's just the hardest to explain. After that, it's straightforward, I think.
There are a lot more papers to go through.
Yeah, we do. But this is just taking the longest to explain.
This is only the stuff from the first exam.
Yep.
So like not even a third of the
I'm not going to do the third part.
Oh yeah.
No, because you already done that. If any of you can set up appointments with me. If any of you want to go over your exam, you can set up appointments with me. But, I'm not Superwoman. I can't do, it forever. Okay, prokaryotic structure. What's the main difference between prokaryotes and eukaryotes? No nucleus. Right. Prokaryotes, no nucleus. Eukaryotes have a true nucleus, and they have organelles, and they undergo mitosis. Which is the biggest? Eukaryotes. Eukaryotes, then prokaryotes, then viruses. Okay? For example, Candida albicans. What's that? Is that a eukaryote or eukaryote? Candida albicans is what causes yeast infections. Yep. So that's bigger than E. coli, and which is a bacteria, gram-negative bacilli, and that's bigger than poliovirus, right? Poliovirus is the smallest. Okay, the shapes. The rod shape is called a bacillus, right? This is a cocci, and that's a spiral. Do I have to write up bacillus, cocci, and spirillum? Okay. So there's three types of spiral.
The circle is the cocci, though.
Yeah. There's three types of spirals. There's vibrio, which is like comma-shaped. Examples of vibrio are cholera. Vibrio cholera. There's spirillum, which is flexible thing. And examples of that is Helicobacter pylori. And then there's the spirochete. Spirochete is, they're long. Examples of spirochetes are Treponema pallidum, which causes,
Sleeping sickness?
you got the S part right. Syphilis, yay! Syphilis. And the other one is Borrelia burgdorferi. What does Borrelia burgdorferi? No! Lyme disease! What's the characteristic sign of Lyme disease? Bullseye rash. Yep. And it's passed by a tick, right? So it's an arthropod-borne thing. I'm getting tired. What do spirochetes have? They have a different type of flagella. Yeah. Did everybody get that? They have an axial filament or an endoflagella. That allows them to snake through vaginal secretions and cause syphilis. Not pleasant to think about, is it? Now, a chain is called a... Yep. Streptococci. Cluster, Staphylo, and you only get clusters with cocci, right? So Staphylococcus aureus under the microscope would be clusters of balls, basically, clusters of cocci, right? If you saw Streptococcus pyogenes would be strings of cocci. Okay? The cell wall. Oh, diplo is pairs. Yeah, strepto is strings. Okay. The cell wall. Gram-positive. Yes. Peptidoglycan. The cell wall of gram-positive bacteria is a large amount of peptidoglycan. What breaks up peptidoglycan?
You said gram-positive equals lysozyme.
A lot of peptidoglycan. Yes, lysozyme.
And then penicillin doesn't break it up.
It prevents the formation of the linking peptides.
What does?
Penicillin. Gram-negatives. They have a little bit of peptidoglycan. So this is the cell membrane here. And then what do they have on the outside?
Yes.
This is the outer membrane. What's the outer membrane composed of?
Lipopolysaccharide.
Yeah, lipopolysaccharide. And there's a very toxic part of lipopolysaccharide.
Isn't it A or something like that?
Yes, lipid A. And what does lipid A cause? Yep. It's another name for it is endotoxin. Causes inflammation. And everybody knows what inflammation is now, right? Fever, vasodilation, shock, blood clotting. Which thing happens first? Vasodilation. Yay!
Vasodilation, heat, and
They all shock due to a drop in blood pressure. And that's caused by the vasodilation, right? Increased blood supply. Clotting, blood clotting. All of those bad things. Lipid A causes it. Lipid A can withstand autoclaving. Not forever, but it can withstand autoclaving. So lipid A is bad, but it's non-specific. Another name for it is endotoxin, right? Because it's part of the cell wall of all gram-negatives. What's the basis of the Gram stain?
The difference in the peptidoglycan.
The difference in the cell wall. Yeah. Yeah. What's the first step in identifying, when you go into the lab, what's the first thing you do in order to identify a bacteria? Gram stain! Yes. The first thing you do is a Gram stain. But there are two types of bacteria that don't have a characteristic cell wall. Have atypical cell wall. One is the acid-fast bacteria. They have a lot of mycolic acid. Examples of acid-fast bacteria are... No.
Mycobacteria.
Yeah, don't get them mixed up. Mycobacteria. And these two types of mycobacteria that we talked about that cause disease. Mycobacterium tuberculosis. What do you think that causes? Macrophages cannot kill tuberculosis, Mycobacterium tuberculosis, unless they're activated.
So TB is what up?
Hmm?
You said there were two.
Yeah, what's the other one? Mycobacterium leprae. What does Mycobacterium leprae cause? Leprosy, exactly. Yeah, that's the one that lives in cool places. That's why it eats off your nose and your fingers. And the current place where the reservoir for it right now is...
What's the other atypical bacteria?
Yeah, I'm going to tell you.
Oh, okay.
There's one other atypical, is what he said. The one that doesn't have a cell wall. Yeah, mycoplasma. What does mycoplasma cause? I told you this right, I think, in the first class.
Walking pneumonia.
Yes! That's the one, that's the dreaded allergy that everybody gets when they go to college, and they're coughing, dry cough, and you never seem to get rid of it, and they're not sick enough to go to the doctor.
Acid fast has a cell wall, it's just...
It's very resistant to Gram staining. So that's why you have to heat it up to get the stain in. Mycoplasma do not have a cell wall. Another name for walking pneumonia is primary atypical pneumonia. So, do you think it normally infects old people?
Everything affected.
Yeah. No, it primarily infects all these college kids.
But everything affected.
Yeah, old people are screwed. Basically.
There's no dignity.
That's right. Okay, the plasma membrane. It's not structural, is it? It's not responsible for the structure of the organism. It's the cell wall that's responsible for the structure. Because if you peel the cell wall away, all bacteria will become spheroplasts. You know, they'll be spherical, like tennis balls. So the plasma membrane is made up of a phospholipid bilayer, right? It contains the enzymes of the electron transport chain in bacteria, which is an energy-like battery. We already talked about that. It's also a permeability barrier that prevents, it lets some things in and other things it doesn't. And then inside the cell, the bacterial cell, you have the nucleoid, which is where the DNA is. And then you have inclusions. We had a question on inclusions. Lots of people got it wrong, didn't they, in the first exam? Yeah, iron and things like that. That's how they store their stuff. They have inclusions. ribosomes are not inclusions, okay? Ribosomes are used to make proteins. And then you often find plasmids. And what do plasmids often carry?
DNA.
Yeah, they're made of DNA. Yes, antibiotic resistance. That's how you select for bacteria having taken up the plasmid. You select for them because it has the gene for antibiotic resistance on it. That's how they pass it between each other so quickly. Okay, external structures. Flagella. Yeah. And they're involved in
Locomotion.
movement. Okay, and if the flagella are all over, what's that called? Peritrichus? Yep. Peritrichus, all over. If there's a tuft of flagella at one end, no. That would be one. If there's a tuft, more than one flagella at an end. Lophotrichus. Do you want me to write that up?
Yeah.
Go on,
Do
you.
it? The first one too?
Yep.
Draw it for us.
Okay, Peritrichus. Lophotrichus. Lophotrichus is like this, right? Peritrichus is all over. And one flagella at the end is monotrichus. If you have two, one at each end, that is, can't remember. Bitrichus. Look that one up. I can't remember.
Just called polar or something?
Yeah. Yeah, I can't remember. I just can't. And then you have the actual filament on spirochetes, which is wrapped around it inside.
What was that third one?
This one here. Monotrichus is just one. Can't remember what
Is it
one is it. What? Amphitrichus, yes! At either end, Amphitrichus. She's hot.
One on each end.
Amphitrichus. I remember Stephanie as Miss Intensity.
No, I'm just aggravated. Like, I was aggravated.
I'm the same way. I was the same way as
I
you.
did not figure it out. This is so bad.
No, it's good. It's good that you're putting that much thought into it. Okay, the little hair-like projections, fimbriae. Yep. And what are they involved in?
Cilia, movement, brain.
Attachment. Cilia are on eukaryotic cells. So they're not involved in movement.
Just
Yeah, don't get that mixed up.
Temporary for sticking and stuff, right?
Yeah,
Okay.
they're part of the slime layer and the biofilm. Okay? And then you can have the pili, they're also involved in attachment. Pili aren't. And then there's the sex pili. It's like the bacteria's little penis. And
Cilia is only in eukaryotic.
what's that? Cilia, yeah. And see, don't get them mixed up. A lot of people get them mixed up with fimbriae because in eukaryotic cells, the cilia are involved in movement, aren't they? Whereas the fimbriae and bacteria are not involved in movement at all. Okay? And the pili are involved in passing plasmids, you know, the sexual conjugation in bacteria, bacterial sex. Okay, the glycocalyx, that's also involved in attachment. That's a sugar coat. And there's two types of glycocalyx. The capsule, what do capsules do?
That's like we saw those. They have like a.
Yeah, and what do they do?
It's
They
protecting it, right?
From what?
from being phagocytized.
Yay! Resist phagocytosis. Makes it much less yummy.
Does anyone have a pen? Mine went out.
How do you spell glycocalyx?
I'll write it up.
Glyco, like.
God, you're making me work hard, aren't you? Is that right? Okay,
Capsule and
slime layer. Yeah, the slime layer is much less organised. It's you know what slime layer is involved in? Biofilm formation. Okay, and the last thing for structure is, and only gram-positive bacteria form these, are spores. Bacillus. Clostridium and Bacillus species form spores.
only gram-positive.
Yeah, they're gram-positive bacilli. Okay? Those are the most resistant biological structures known, are spores. They contain dipicolinic acid. Bacteria make spores when they run out of nutrients, right? And spores are a great concern for biological warfare. Biological warfare. Which microorganism makes spores that we're very concerned about? For anthrax, yeah. Bacillus anthracis. Most of these Clostridium, those are all anaerobic. Okay? But if you remember those two genuses, Clostridium and Bacillus, then you'll remember that they're the only ones that make spores. Okay? That will help you remember them. Okay? Okay, where they grow, microorganisms grow. Do I need to remind you of this?
Yeah.
I have to remind you about every bloody thing, don't I?
Oh, some of them are at the surface, some are at the bottom, some are like equally.
Yeah, so this will be aerobic, right? We're talking about growing in both.
Some are pyrobic.
This is facultative anaerobes, right? What can facultative anaerobes do? They can grow either way.
But everyone prefers oxygen.
But they prefer oxygen, that's why you're going to see more at the top. Okay? So examples of facultative anaerobes are like the yeast that are involved in making beer and wine. If you're not getting enough alcohol, you've got too much oxygen present, right? Because you don't get, they prefer to grow in the presence of oxygen, so they won't make alcohol if there's oxygen around. Did everybody see that? Remember there was a question of that on the exam. Okay, so then the obligate aerobes will be growing at the top. Obligate means it can only grow in the presence of oxygen. So we're obligate aerobes, right? Okay, and then the obligate anaerobes, they'll only grow at the bottom, right? Obligate anaerobes. And then microaerophilic will grow right here. They can withstand a small amount of oxygen. Microaerophilic are strep pyogenes. It's microaerophilic. And the aerotolerant, they don't use oxygen, but it doesn't hurt them, so they just grow throughout. Do I have to write up each
What was that
time?
one?
Aerotolerant.
So,
And this one's microaerophilic. Yeah.
so the obligate aerobic require oxygen. The facultative anaerobes, they can do without, they can live with it, without it.
But they prefer to, they grow faster with it. Yeah.
And then the obligate anaerobic, they have to live without it.
Yeah. They can't live in the presence of oxygen. You got it?
That's like kind of towards the top for the micro one.
Yeah. Just below the surface. They can get rid of some of the toxic oxygen. Helicobacter is another one that's microaerophilic. Okay, the growth phases. Do you remember the growth phases? Do you want me to go over the growth phases? Yeah. Yeah. What's this called?
Mag.
Mm-hmm.
Oh, the lag log.
Okay, this is logarithmic or exponential, and this is logarithmic or exponential.
You don't have to explain that, I guess.
What about everybody else got this?
Yeah.
Do you want me to explain it? Good. Okay. Spores are killed by hot air sterilization, and they're also killed by the incinerator.
Go by what kind?
Hot air sterilization, any sort of sterilization by definition will kill spores. That's the definition of sterilizing something. So, and you can kill them by incineration, hot air sterilization, and the classic way to get rid of spores is the autoclave.
But can't something survive out of living?
Yes, prions. But I'm probably the only one who taught you as much about prions.
We were just talking about something. Oh,
Spores.
Endotoxins survive out of living.
Yeah, it does, but it's not alive.
Yeah.
Yeah. Yeah. Spores, determines alive bacteria. Okay. The autoclave, you do 121 degrees for 15 minutes. Fahrenheit. Okay. If you don't have an autoclave to sterilize things, what's another way? Something else you can use that would do it also?
Pressure.
Yes, pressure cooker.
Oh, really? That's right. Okay.
Yeah, pressure cooker. As long as you did it at 121 degrees for 15 minutes. Because an autoclave is like a pressure cooker. It's a more sophisticated version. Disinfection is destruction of the vegetative forms of organisms on an inanimate surface.
Wait, say that again?
Right on.
No, like.
You remember disinfection,
Yeah.
Just
You're
sterilization.
destroying all the vegetative forms, so you're not destroying spores. Remember?
He's putting it down to a safe level.
No, you're actually destroying them all with disinfection. On an inanimate surface, like the table.
Sterilization is when you're going into a safe level.
And no, sterilization is when you kill everything, including spores. Okay. Disinfection is when you're killing everything but the spores on a surface like this. So you're killing the vegetative forms. Because remember, when a spore, when it turns back into a bacterial cell, it's called a vegetative cell. Right. Okay? So when you're destroying all the vegetative forms on human tissue, on living tissue, or animal tissue, what's that called? They do that before they do surgery.
Antiseptic.
Antisepsis. Do I have to write that one down too? So, you know, when they put betadine on you before you're having surgery, that's antisepsis, right? Because they're killing everything off living tissue. But they, you know, both disinfection and antisepsis do not get rid of spores. And sanitization is reduction of the microbial population to a safe level.
Sanitation.
Yeah, sanitization. You know, so skin sanitizers, they have to, by definition, remove 99.99% of bacteria. So there are still 0.01% left. And if you think about that, that's actually quite a few. Because remember, it doesn't take long for one bacteria to become a lot. So by definition, that's what skin sanitizers do. So it's just reducing the level. Examples of sanitization are washing hands, washing your utensils for eating. Pasteurization, as I said, reduces heating to a 63 degrees for 30 minutes, which gets rid of most of the pathogenic bacteria.
To what degrees?
It's 60, you don't have to remember that.
Oh, okay.
63 degrees for 30 minutes.
Celsius.
Yeah, no, Fahrenheit. This is America.
That's enough to.
60-something degrees, that's not even hot.
Oh, it must be Celsius then.
Yeah.
Yeah, because this is gonna be
That's gonna be
Celsius too.
Celsius too.
Yeah, because in science, you use Celsius. Yeah, yeah. 63 degrees Celsius. Yep. Sorry. Made a mistake. I don't make many. Yeah, I do really. And then there's desiccation, radiation, and filtration. You filter things.
Is that you, Steph? Is that you?
Mm-mm.
Oh. Yours got your shirt on?
It's my painter.
No, making sure it's recording.
But I can't answer it right now. Okay, so
Pay the house phone for it.
what's that?
In the house phone.
So filtration, if you can't autoclave things, like proteins can't be autoclaved because they lose their secondary structure and tertiary structure, so you filter them. You can get rid of bacteria by filtering them. Okay, radiation can get rid of things that you can't sterilize too, and desiccation is just drying them, like salting out, and you know, drying can kill a lot of bacteria. For chemicals, the halogens such as chlorine and betadine, iodine, they're very good. The surgical scrub, the two types of surgical scrubs are chlorhexidine and betadine.
Is it chlorhexidine?
Yeah, and betadine. Those are the two surgical scrubs. Okay. Phenolics and alcohol, and then we have surfactants such as soap, you know, dissolved membranes. Surfactants dissolve membranes. And heavy metals, such as copper and silver nitrate, copper sulfate and silver nitrate. And then for gases, we have ethylene oxide. That can sterilize things. In fact, they use that. Okay, talk about viruses. God, I'm tired. Ugh, how much more to do? Got that? Can I start talking about viruses? Viruses consist of, the basic structure of viruses is very simple. A nucleic acid, they either have DNA or RNA, but never both, right? As opposed to bacteria, which have both. We have both too. And then you have outside of the DNA or RNA, you have surrounding that a thing called a capsid, which is there to protect the nucleic acid. It's composed of proteins, okay? And the proteins are called capsomeres. And the envelope they don't always have an envelope. If they have an envelope, they're called envelope viruses. If they don't, they're called non-enveloped or naked. Yeah. So you can have an envelope outside this. Where does it get its envelope from?
The outside of the cell when it breaks through.
Yeah, when it's budding. Yeah, it gets it from the host cell membrane. The envelope comes from the host cell membrane. Okay, and then outside that it can have spikes. But they don't always have spikes. They're made of glycoprotein.
The spikes are?
Yeah. An example of a virus that has spikes is influenza. Remember, influenza has two types of spikes, hemagglutinin and neuraminidase. I'll talk about that more in a minute. Okay. Do viruses contain peptidoglycan? No. What contains peptidoglycan?
Bacteria.
Bacteria. Everybody's becoming punchy, and I am too. I can tell. Okay. And they have that, because these proteins fall into these viruses self-assemble. The capsid can have that structure, which is called a icosahedron, which is 20 triangles, because that's what naturally forms. It's like a diamond, looking like a diamond. An example of that is poliovirus. Poliovirus is a naked virus. Herpes
Meaning it doesn't have an envelope?
Yes.
Or spikes?
And an envelope virus is herpes. Okay? You can have helical viruses where the capsids, it's like this. Where the capsids. An example of that is Ebola. Okay? Ebola, they're having an epidemic of Ebola in Africa right now. 140 people have died of Ebola. 142, sorry. Terrible disease.
Question,
Yeah.
the exams, about like the prevalence of it.
I won't say the prevalence of Ebola. I'm just giving you that information.
It's easy to remember, of course they're going to ask.
So the life cycle of a virus. The first step is, it's got a stick, doesn't it? What's the stick called?
Adherence.
What's that?
Adherence?
Yeah, what's in, but in
Well, you
virus
just tell us at
language.
the same time.
In person.
No, adsorption.
Oh, yeah.
Absorption.
Yeah,
If you're absorbing something?
yeah.
Okay.
Sticking. They got to stick in order to do their thing, right? After it's adsorbed or stuck, what happens next?
Is it
Injection.
Yeah.
Absorb or ab?
Ad.
Okay. Sorry, I just can't tell.
I know, it's my accent. Okay, what's the next step?
Injection.
Yeah, and what's that called?
Perforation.
Penetration.
Not a good enough reason to use that word.
Yeah, this is scientific lingo we're using here. So it's not going to be injection.
Right, penetrate is so much better.
Penetration, yeah. Penetration, what comes after that?
Then they
Replication, I don't know.
That's with bacteriophages. Uncoating. They've got to uncoat, right, don't they? Sometimes they uncoat as they penetrate.
I don't think so.
And then what happens after it uncoats?
So the next one was
Bunch of
uncooked?
movie makers.
Yeah. Then it has to be replicated, doesn't it?
Well, that sounds.
I thought it couldn't be replicated, or they can't replicate themselves.
No. Yeah, they can't replicate themselves. They get the host cell to do it for them. So the next stage is replication. I'm giving you the simplified version.
Appreciate it.
In bacteria, it's the injecting part here. It's not actually called injecting, but you got the right idea. Okay, and then after that, after replication, you get all the parts made, so you've got to join them all. Assembly! Stephanie's, I told you, Stephanie's hot. So you get assembly, and then release, right? And with mammalian viruses, there's two ways of release, right?
Budding.
Yep, budding. Budding only occurs with envelope viruses, right? And if they bud, the cell doesn't die. Well, it can die, but not necessarily. Okay, and the other way is to lyse the cell. Just blast out. So bacterial viruses all do that. They blast out. Okay? So naked viruses kill the cell. They lyse the cell. With the absorption of an envelope virus involves fusion of the viral envelope with the cell's membrane. So the envelopes fuse with the cell membrane, okay? Latent infection, that's
The equivalent of the lysogenic cycle in bacteria.
very good. Latent infection is when the virus stays inside the cell. And that's in mammalian cells. Which type of viruses all become latent?
Herpes?
Yeah, retroviruses do too. Herpes. All the herpes viruses become latent. Sorry, once you have them, you never get rid of them. You've got them for life. You're all stuck with them.
Thanks,
Okay.
Sarge.
You may not want to believe that, but it's true. And the lysogenic cycle in bacteria is equivalent to this. What's a bacterial virus called?
Bacteriophage.
Yeah, bacteriophage, right? So the lysogenic cycle is when the virus DNA becomes part of the bacterial DNA. And then the bacteria is said to be lysogenized. So if the bacteria is lysogenized with a particular bacteriophage, it can no longer be infected by viruses of that type. Okay? It's immune to being infected with any more bacteriophages of the same type. However, it can be infected with other types of virus. So it's not immune to other types of bacteriophages, but it's immune to being infected with the same type of bacteriophage. Does everybody get that? Okay, the primary infection, if you get an infection that happens very quickly and you get very sick very quickly, what's that called? It's an acute infection. It comes on rapidly and you get very sick and then you either die or get better. Infections that carry on for a very long time, they're called chronic. The type of infection is usually due to how fast the virus or bacteria can replicate. As I said, all herpes viruses become latent. Herpes simplex is latent in your trigeminal ganglion, which is right here. Okay. Are viruses killed by antibiotics?
No.
No, only bacteria are killed by antibiotics. Retroviruses, what do all retroviruses contain? What enzyme do they all contain? Yeah. They're all, part of their life cycle is to become latent in the host cell's DNA. They become part of your DNA. That's why once you
Reverse transcriptase.
Yeah, that's why once you become infected, your cells become infected with a retrovirus, unfortunately it's there for life because it becomes part of the cell's DNA. Retroviruses cause tumours and AIDS, right? Hepatitis B also becomes latent because it also has reverse transcriptase. Hepatitis B. Which chemical kills virus-infected cells? That was one of the questions in the exam.
Is it interferon?
Yes.
I got that.
Interferon.
What chemical kills what?
Virus-infected cells. Interferon.
That's like the last dying whatever, right? Oh no, that's just like,
Doesn't it just kill itself because the other cells that are around it from also getting it?
It prevents the other cells, yeah. Okay. Herpes viruses.
what is that?
Herpes viruses. Different types of herpes viruses.
Yeah.
Herpes simplex type 1 causes cold sores. Type 2, sexually transmitted diseases. The type 2 becomes latent in the lumbar sacral nerve, but these days type 2 causes cold sores and type 1 causes genital herpes.
But if you get one, you can't get the other in the same area,
I don't think so.
right?
But if you get type 1 here, I don't think you can get type 1 down there.
That's what.
If you
Oh,
get
yeah.
type 2 down there, I don't think you can get type 2 up here.
You can still get type 1.
Correct?
Gross.
Where did you say, simplex 2 was latent?
Trigeminal ganglion. Oh, 2, lumbar sacral nerve. But it's both ways now because of all your bad behaviour of young people. Herpes zoster. What is that called?
Chicken pox.
Yay! And what is the reoccurrence called?
Shingles.
Yay! And it becomes latent in the dorsal root ganglia. You get an itchy rash, fever. It can cause fetal damage. There's a vaccine for it. Treated with acyclovir breakthrough, give vaccine to 60-year-olds to prevent shingles.
Is the rest of this review gonna be on different infections?
Yep.
Because I think I got those locked down. And if so, then I will see
Yep.
The rest of it's infections?
Yep.
Herpes and
Yeah.
Everybody wants to stop? I'm happy to stop.
Yeah, no, that's useless.